Hey guys, welcome back. It's Adam the Science Tutor. Today we're going to be talking about gluconeogenesis, and the purpose of gluconeogenesis is to make glucose from glycogen or from amino acids or oxaloacetate or from pyruvate. Specifically, we're going to be talking about getting it from pyruvate. But what do we need glucose for? Well, glucose is essentially needed for energy. It's a low energy form. We store it in glycogen. So we need to either break up the glycogen or we'll go through gluconeogenesis during the bad times where we may not even have glycogen storage, and our brain is always using glucose, so we need glucose. So gluconeogenesis is for the creation of glucose. It's almost the opposite of glycolysis, because if you think of glycolysis, we take glucose and we could either go through the pyruvate shunt, or we can go through glycogen synthesis, or we can go down and make a direct form of energy, ATP and whatnot. So it's pretty much the reverse form of glycolysis. And if you guys check out my glycolysis video, you'll see that the enzymes and all the products and byproducts of glycolysis, so I'll put a link to that at the end of this video. And all enzymes are the same, and they're all reversed except for the key regulatory enzymes of glycolysis. I don't know if you guys recall, but the key regulatory enzymes of glycolysis are hexokinase, phosphofructokinase, and pyruvate kinase. So all three are kinases. And if you don't remember it, it's okay. I drew out glycolysis and we'll get looking at it. But all other enzymes in glycolysis are the same in gluconeogenesis. It's just reversed. It's going back up. They're all reversible reactions, except for the key regulatory enzymes. And key regulatory enzymes are not reversible. So these three are going to be the difference, are going to be different than what glycolysis is. But if you have glycolysis memorized, then kudos to you. You have most of gluconeogenesis memorized as well. So this is a glycolysis, and I didn't do reverse arrows, but I'll draw them in a bit. But we have our hexokinase, phosphofructokinase, and pyruvate kinase, our three key regulatory enzymes of glycolysis, which are going to be changed for gluconeogenesis. Over here, I'm drawing a little key for you guys to talk about it a little bit in glycolysis, but these are key enzyme names. So kinase is like a direct energy. Isomerase or mutase is for conformation changes. And dehydrogenase is like an indirect form of energy. So direct form of energy is like ATP, ADP, AMP. The indirect energy is NADH or FADH2, which can be broken down to 3 or 2 ATP molecules, respectively. So pyruvate kinase is the first enzyme that we need to change, or that we needed to show. I'll show you how to get from pyruvate to phosphoenolpyruvate. So in the mitochondrial membrane, we're going to have our pyruvate molecule getting ready to go through the Krebs cycle or traveling to the Krebs cycle. So that's where our pyruvate's at. And so pyruvate, we're going to need to change that to oxaloacetate. And oxaloacetate is another molecule in the Krebs cycle. It's essential in the Krebs cycle. And so what does this? Well, we need to put a carbon dioxide on the pyruvate. So an enzyme helps us with the name. It's called Pyruvate Carboxylase. And so we're going to put a carbon dioxide molecule on the pyruvate. So we lose carbon dioxide, and we're going to form oxaloacetate. And oxaloacetate is a important or vital portion of the Krebs cycle. So, but oxaloacetate cannot leave the inner mitochondrial matrix or membrane. Remember that the inner membrane of mitochondria, got stick-tongued, is where the Krebs cycle occurs. So we want to keep the oxaloacetate there. Therefore, we're going to have to use the reversible reaction and put a carbon dioxide on the oxaloacetate to make it into malate, which is a reversible reaction and a part of the Krebs cycle as well. And malate can now get distributed to the cytosol, which is where we want our oxaloacetate. So how will we now make oxaloacetate now that we're in the cytosol where we want to be? We'll Just like the Krebs cycle, malate dehydrogenase will occur. Dehydrogenase, if you remember, is an indirect form of energy, so we'll get NADH as a product, and we need an NAD molecule in the cytosol to make our oxaloacetate. And yes, this is very redundant, however, it's important, so we go from oxaloacetate to malate back to oxaloacetate. But no new real enzymes, because these are mostly used in the Krebs cycle. So if you know the Krebs cycle and glycolysis, you're so far should be good. So we're going to take oxaloacetate and we're going to try to make it to pyruvate, or no, phosphoenol pyruvate, which is going up the ladder of glycolysis, if you guys remember. And this is done with the enzyme phosphoenol pyruvate carboxykinase, which is a whole mouthful. So phosphoenol pyruvate carboxykinase. And if you break down that word, carboxy means the oxaloacetate is going to be carboxylated or get a CO2 molecule, and kinase means that we're going to get a direct energy or lose direct energy. But in this case, we're going to lose the CO2 and attach it onto the oxaloacetate, and we're going to gain an ATP, a direct form of energy. So cool. And now we have our phosphoenol pyruvate, which is moving up the ladder of glycolysis, so we're one step closer to beginning our glucose molecules. And phosphoenol pyruvate, how did we get that? Well, we had our 2-phosphoglycerate, and amylase helped make our phosphoenol pyruvate, so that's going to keep going down. This is important, negative inhibition by ADP, which makes sense. So it's negatively impacted by ADP production. So now we're going to go up the ladder. So we use enolase to go back up, phosphoglycerate mutase to go up, phosphoglycerate kinase to go up, glyceraldehyde 3-phosphate dehydrogenase to go up, amylose to go back up, and now we're next to our next key regulatory enzyme of glycolysis, meaning we're going to need another pathway. We can't just use the glycolysis pathway. So what can we do? Let's look. So we have phosphofructokinase, so we're going to go from 1,6-fructobisphosphate to fructose 6-phosphate, so that we can continue going up the ladder until we hit our last key regulatory enzyme, and then we'll have finally glucose, which is where we want to be. So we have our 1,6-fructobisphosphate, and then we're going to use an enzyme, and we kind of already know the enzyme names, so we're probably going to have 1,6-fructobisphosphate in the name to make fructose 6-phosphate. So what do you guys think it could be? Would it be carboxylated? Will it be kinase? Well, let's see. So, and this one is probably the hardest one to say, but these next two, but it's an easy pathway change. So we have fructose 1,6-bisphosphate and bisphosphatase. So fructose 1,6-bisphosphatase, or you could say F-fructo 1,6-bis for 2 phosphate, and then tase because we're gonna get rid of a phosphate. So we're getting rid of a phosphate, and we're gonna create fructose 6-phosphate. And it's negative inhibited similarly by AMP, adenine monophosphate, and it's increased with citrate, which makes sense. So cool. And then we're going to go up the ladder, continuing up. We're going to go from fructose 6-phosphate to glucose 6-phosphate by glucose isomerase, and it's just reversed. So most of these are reversed. So once again, it's similar to glycolysis, just going in reverse, except for these 3 enzymes. So we're going to get glucose 6-phosphate and convert it to our D-glucose, which is our glucose that we want for our body. So water is going to be inputted, and similarly named, it's going to be G6 glucose 6-phosphate tase. So we're also going to remove a phosphate group, which makes sense. So glucose 6-phosphate, or phosphatase, my bad. So glucose 6-phosphatase to make our D-glucose, which is ready to be used as immediate energy for our brain or muscles. So yeah, the last 2 key regulatory enzymes are easy reverses. They're just one step. The hard one was the pyruvate kinase for sure. But once you memorize that redundant step where you go from oxaloacetate to malate to oxaloacetate just to get it out of the cytosol, then you should be set. So yeah, so where does this usually happen at? Well, gluconeogenesis occurs primarily in the liver and in the kidney. And if you can remember what's important about kidney and liver, what's their function? Well, they make a lot of blood for sure. They do other things as well, but they make a lot of blood. So gluconeogenesis occurs 90% in the liver and 10% in the kidneys. So, and that makes sense. And we want this to happen next to the blood. Why? Because if our brain is low in glucose, it means we're having brain damage. We need the glucose next to blood so it can be taken up to the brain as quickly as possible. And that just is the way it makes sense. So thank you guys for watching and tuning in. Please check out next videos or the other ones. It's Adam the Science Teacher. Appreciate it.